Start with this idea. For decades, scientific breakthroughs, well, they've been driven by researchers looking at petri dishes, running endless trials, relying on, expert intuition. But what if the most cutting-edge discovery in a field as complex as cancer therapy wasn't found through that kind of brute force human trial and error? What if it came from a machine, one capable of generating a totally novel, testable, and scientifically valid hypothesis? Hi, I'm Edward, your co-host today.
And I'm Lily. We've got a great deep dive today, so let's jump in.
Welcome back to the Deep Dive. Today we're focusing on what feels like a landmark collaboration. It's between Google DeepMind and researchers at Yale University. They deployed this, well, absolute behemoth of an AI, a 27 billion parameter foundation model, and they use it to tackle one of the most stubborn puzzles in modern oncology. It really feels like a case study proving AI isn't just sorting data anymore, it's becoming a critical partner in discovery.
Yeah, the central insight we're exploring today is truly profound. This AI model called C2S scale 27B, it successfully navigated this incredibly vast complexity of cellular interactions. And it identified an existing drug, silmitasertib, as what they call a conditional amplifier. Crucially, it predicted that this specific drug, but only when combined with low-dose interferon, could flip the switch needed to turn previously cold invisible, really tumors hot.
Right, making them visible to the immune system. And the real weight here, the reason we're doing this deep dive, is that the AI's complex, pretty nuanced prediction. It was fully confirmed in the lab. This isn't just theory. It's a new, proven therapeutic pathway.
So our mission today is clear. First, we need to set the stage, understand the biological challenge, what actually makes a tumor cold. Then we'll dig into how this AI was, well, fundamentally trained to speak the language of a cell, which sounds kind of wild. And finally, we'll reveal the specific, non-obvious, synergistic interaction the model uncovered, giving you the core knowledge behind this landmark discovery.
Okay, let's unpack this. We absolutely have to start with the problem, the thing that prompted this massive undertaking, this difference between hot and cold tumors. It's arguably the single greatest obstacle in modern immunotherapy, right?
So absolutely. The success of immunotherapy, which, just as a reminder, teaches your own immune system to fight cancer, it's almost entirely dependent on whether that tumor is visible.
So let's define visibility.
Yep.
The hot tumors, they're the ones we want. These tumors are highly visible because their cells are actively presenting antigens on their surface. Think of antigens like little flags saying, hey, I'm foreign, attack me.
Exactly. And the mechanism for displaying those flags is key. It relies on high expression of this complex protein machinery called MHCII. If the tumor cells have high MHCI expression, then the T cells, the immune system's specialized soldiers, can recognize them, lock on, and destroy the cancer. So if you have a hot tumor, immunotherapies like checkpoint inhibitors, they often work incredibly well.
But then you just hit a wall with the cold tumors.
Yeah.
And these are the majority of cancers that resist immunotherapy. They're, well, frustratingly invisible.
Right. They are essentially cloaked because they exhibit low MHCI expression. It's like those cancer cells have just ripped off their ID badges. They exist in what scientists call an immune contact neutral environment. They just hide, they evade detection, and that makes most of our advanced treatments pretty ineffective. They're really the central reason why so many patients don't respond to some of the most promising cancer treatments we have today.
So, okay, if the goal is to make these cold tumors hot, researchers need to find the specific lever, a drug, or maybe a combination of drugs, that forces those cancer cells to dramatically increase their MHCI expression. But that sounds like a massive chemical search problem. Thousands of variables.
And that search problem is simply too complex, really, for traditional methods. You can't just manually test every permutation of every existing drug against every possible cellular state. The sheer scale, the nuance of the cellular environment, it just screams out for a computational solution.
Which brings us neatly to the tool they used. How do you even build an AI model that can not only read all of biology, but also reason about these subtle chemical interactions within that environment?
Well, this is where the sheer scale of the cell-to-sentence scale 27B model, the C2S scale, comes into play. It's built on the foundations of the large language models we're all getting familiar with now, like Google's Gemma family. But instead of predicting, say, the next word in a sentence, it predicts the next biological state of a cell.
27 billion parameters. That number is hard to wrap your head around. It just suggests massive complexity. What about the training data? How do you train an AI on biology as language?
So the core innovation here is what they call cell-to-language translation. Instead of text scraped from the web, the model was pre-trained on this vast multimodal corpus, over 50 million single-cell gene expression profiles. These profiles are generated using something called single-cell transcriptomics.
Okay, hang on. Let's pause on that term. For listeners who aren't geneticists like me, what exactly is single-cell transcriptomics?
Right, good question. So think of the genome as the cell's master blueprint. It's mostly static, right? Transcriptomics, on the other hand, measures all of the messenger RNA, the mRNA, being produced in a single cell at a specific moment. It's like the cell's dynamic moment-to-moment active reading list. It tells you exactly which genes are turned on, how loudly, and what the cell is actively doing right now. Is it resting, dividing, hiding from the immune system?
Got it. So you fed the model the detailed real-time active reading list of 50 million individual cells, showing how they behave under thousands of different conditions, different stressors.
Exactly. And this allows the AI to essentially read the genetic language of these complex cellular states. It can then translate that complex gene expression data into, well, sentences that it can analyze, infer meaning from, and reason about. It learns to understand that low MHCI expression is basically the cell saying, I am cloned.
What's really fascinating here, and the sources stress this, is the significance of the scale. We know that in human language models, capability scales dramatically with size. More parameters mean better coherence, better handling of complexity. Did those same scaling laws actually hold true for biology?
They did, and that's frankly a landmark finding in itself. This discovery really demonstrates that the complexity of cellular language demanded this massive 27 billion parameter model. A smaller model might find some obvious connections, sure, but the subtle stuff, the nonlinear interactions, the ones that kind of defy simple logic, those require that enormous capacity for nuance. The scale was absolutely essential for detecting the hidden synergy they were looking for.
Which leads us directly to the very specific job they gave this highly trained biological oracle. They didn't just ask for any solution, they asked it to find a conditional amplifier. That phrase feels dense with meaning.
It really is, and it speaks to the high level of sophistication they needed. The human researchers didn't just want a drug that boosts antigen presentation everywhere, all the time. That kind of approach is usually pretty toxic, non-specific. They wanted a drug that provided leverage, but only under specific beneficial conditions.
So the condition was everything. What were the two conditions the drug absolutely had to meet simultaneously?
Okay, first, the primary goal. It had to significantly boost antigen presentation. That's the visibility boost we talked about. But second, the crucial conditional part. It had to only exert this strong effect in an environment where low levels of interferon were already present.
Why the specific focus on low interferon? That seems incredibly precise.
Well, because interferon is a natural immune signaling protein. Low levels are actually often present in the tumor microenvironment, but they're just not high enough on their own to trigger a full immune response. The researchers had this hypothesis. If they could find a drug that amplified that existing low signal, they could selectively turn the tumor hot without, flooding the whole body with high-dose, often toxic interferon. It was really a search for the perfect molecular co-pilot.
That's incredibly elegant looking for sort of the weakest link in the system that could be leveraged.
Right.
Okay, so how did the AI actually go about finding this specific molecular co-pilot among, thousands of available compounds?
The C2S scale model conducted what's called an in silico. That just means a computer simulation dual context virtual screen. It systematically tested the predicted effect of over 4,000 existing drugs, and it modeled the outcome of each drug across two distinct simulated environments. One with low interferon and one completely without it.
A massive virtual experiment. And what did this complex screening process reveal? What was the AI's novel hypothesis?
So the C2S scale model singled out the drug silmitasertib, which is also known as CX-4945.
Yeah.
It identified it as this powerful conditional amplifier they were looking for. Pharmacologically, silmitasertib is known as a kinase CK2 inhibitor.
Okay, let's quickly clarify. What does a kinase inhibitor actually do?
Right. Kinases are really essential enzymes. They act like molecular switches inside cells, turning processes on or off, usually by adding a phosphate group to other proteins. Silmitasertib inhibits one specific enzyme, kinase CK2, so it essentially jams one of the cell's key regulatory switches. The AI hypothesized that jamming this specific switch was the keys to unlocking the pathway needed to leverage that low-dose interferon signal.
And the really non-obvious part here is the synergy, right? It wasn't just predicting that this inhibitor was, like, generally good for the immune system somehow.
Precisely. That's the core novel prediction. It wasn't just about one drug or the other. It was that three factors were required for a meaningful outcome. Silmitasertib alone, negligible effect predicted. Low-dose interferon alone, also a negligible, maybe very mild effect. It was the precise combination of the two that the model predicted would unlock a dramatic synergistic increase in antigen presentation. No human analysis had previously flagged this specific pairing as having this kind of powerful conditional effect.
Wow. That's the scientific gold standard, isn't it? An unpredicted, complex synergy. Okay, so once the machine generated that hypothesis, the weight of proof shifted back to the human researchers at Yale. How did they go about validating this in the lab?
They immediately moved to an in vitro validation phase. That means using human neuroendocrine cell models in the lab. They systematically tested the predicted effects, the drug alone, interferon alone, and then, crucially, the combination.
And did the real-world biology, in the dish confirm the machine's prediction?
Stunningly, yes. The experimental results mirrored the computational model almost exactly. When silmitasertib was applied alone, they saw no significant change in MHCI expression, just like the AI said. Interferon alone resulted in only a very marginal change.
But the combination, did it achieve the goal?
The combination achieved what has to be called a landmark result. The pairing of silmitasertib and low-dose interferon caused a really remarkable, approximately 50% increase in antigen presentation. They had successfully forced these formerly invisible tumor cells to express their ID badges, confirming a brand new biological pathway that makes cold tumors visible and therefore potentially targetable.
50%. That is a massive jump in visibility. It's a critical data point that validates this entire approach, doesn't it? This really shifts AI from being just an assistant to being maybe an originator of scientific theory.
It feels like the ultimate proof of concept. For years, we've used AI to categorize things, sort data, identify patterns and stuff we already kind of knew was there. Now we have an AI that, through its ability to understand biology as language, has generated a complex, multi-factor hypothesis. And that hypothesis was proven correct in the lab, establishing a genuinely new therapeutic path.
It almost redefines the role of the scientist, in a way. Instead of running thousands of blind experiments, they're now maybe focusing on validating the top hypotheses spit out by the machine.
Exactly. And the significance here extends far beyond just this one drug combination. I mean, while this isn't a cure yet, it obviously needs much more clinical testing, it opens a really crucial door. A door for developing advanced, highly specific combination therapies that could potentially work for that huge group of cancer patients who currently have cold, unresponsive tumors.
And one of the most exciting aspects for the broader scientific community is Google's commitment to actually open source this knowledge, right?
That's right.
Yes, absolutely. They've made the C2S scale 27B model and its resources available. This means the entire scientific community can now potentially use this powerful tool. They can use it to accelerate future research, testing other AI-generated hypotheses, probably in fields far beyond just oncology. It dramatically lowers the barrier to entry for this kind of sophisticated hypothesis generation.
That accessibility really implies that the pace of discovery in complex systems, like the human body, might be about to experience a profound acceleration.
It definitely feels like a potential game changer. We are witnessing this fusion of computational scale and deep biological complexity.
So as we wrap up this deep dive, let's just quickly recap the journey. We started with that massive challenge, cold and visible tumors. We saw how the C2S scale 27B model, trained on, 50 million cell profiles, was built to essentially speak the language of cellular biology. And we finished with the successful, really non-obvious discovery of silmitasertib plus low-dose interferon, a synergistic combination predicted by AI and confirmed to boost tumor visibility by that remarkable 50%.
Yeah, and I think the biggest takeaway for you, our listener, is maybe to consider what this implies for biology itself. If scaling these models allows AI to truly unlock complex biological language, treating the interactions of genes and proteins not just as chaotic data points, but as an interpretable, scalable kind of syntax, then what other foundational mysteries in human health might be next? This ability to reveal previously hidden synergistic pathways, it fundamentally changes the role of technology. It feels like we're moving into an era of machine-aided scientific intuition, an intuition that could solve problems we haven't even been able to properly formulate yet.
A shift from just sorting data to actually generating true true scientific insight. It's fascinating stuff. And hey, if you enjoyed this deep dive, please do consider liking and subscribing wherever you get your podcasts so you don't miss our future explorations into fascinating topics.
That helps us out a lot.
You can also find the latest articles, full transcripts of our discussions, links to partner products we sometimes mention, and more over at www.dansplain.xyz.
Yep, check it out.
We'll catch you next time.